Netcasts you love.
From people you trust.
This is Twit.
Bandwidth for Futures in Biotech is provided by CashFly at cashfly.com.
This is Futures in Biotech, episode 60. You come to this cave often? This episode of Futures in Biotech is brought to you by Audible.com. To download a free audiobook of your choice, go to audible.com/biotech.
I believe that biotech is the next frontier.
Probably the greatest intellectual revolution that's ever taken place in man's history. DNA is the code for life. We're actually beginning to understand how life works, which I think is something that's mind-blowing in and of itself.
There was going to be a genetic component to aging.
How long was there the extension?
About 30-40% for humans. That would equate to something like 20 to 30 years.
How close are we to actually having a therapy of some kind?
Ballpark, 10 years.
It's potentially one of the things that'll end up rocking the world the same way that people said, oh, the sun's the center of the universe, oh, this and that and everything. And now here's somebody who can come out and say, hey, look, here's how we compare it to our closest evolutionary relative.
All right, so let's start the show. It's Futures in Biotech, episode 60. And since it's a kind of a round number, we've decided to do a panel show. And which we do about every 6 months. And what it helps me do is collect what are the most important scientific stories that are happening this year. And I then go out and contact the authors of these papers and invite them on as guests. So I've invited a number of familiar people. We have Dr. Dave Brodbeck, who's an associate professor and chair of the Department of Psychology at Algoma University. Are you associate dean there too now?
Oh, no, I'm the head of the Division of Science as well. Interim.
And you're obviously a Habs fan, which is really cool.
Habs go.
I approve.
Exactly. So I think that's fine. Poor Pittsburgh people and Philly people. Okay, but they've been doing pretty well. Andre, that was the voice of Andre Nantel, Senior Research Officer at the National Research Council of Canada. We've got now on the screen, we have Vincent.
That's not me.
Nope,
Man, I've gotten ugly.
Oh, man. Well, we're going to have a scientific debate here. Okay, there's Andre. And that other person we had on the camera was Vincent Ragniello. He's professor of microbiology and host of This Week in Virology, the very popular podcast. You can find it on iTunes under medicine. It's always featured, so it's really great to have him on the show. He's also the host of This Week in Parasitism.
Hey, Mark, good to be back. Haven't seen you in ages.
It's been a little while. You were the co-host of a show with Carla Kierkegaard recently, and that was a really fun show.
Too. Yep. A lot of fun.
Oh, yeah. Dick de Pommier as well. We did last episode that aired, right, or 2 episodes ago. Vincent Ragniello, he's professor at Columbia University. And last but not least, we have Dr. George Farr, who's Vice President of Biochemistry and Biophysics at Aromics LLC in Cleveland, Ohio. Welcome to the show.
Hello.
Sitting right beside you.
Yeah.
So I was thinking we should, we don't have that much time, but we have some really interesting stories. I have one PDF document that I'd like to send. Can I send it to you, Burke, by email, or should I send it to you by Skype? File transfer. I can't hear him. Anyway, it's not that critical. Don't worry about it. Okay. Do I email it to burke@twit.tv?
Yes, you do.
Awesome.
Bring on the spam.
All right, so let's start talking about the first story, and I thought we'd start with Dave Brodbeck because he's got a really wild story, and he's got a good handle of it. So, how about you tell us a little bit about what's going on?
Thanks, Mark. I don't know how much of a handle I have on it, but it's, to me, a cool paper because most of the things I've ever talked about in the show are rather sort of molecular neuroscience kind of things, whereas this one is purely behavioral. And the paper's called Thatcher Effect in Monkeys Demonstrates Conservation of Face Perception Across Primates. And it's by Adachi, Chu, and Hampton. And Rob Hampton's an old buddy of mine from grad school. Basically, in this paper, and hopefully if Bert gets that PDF and pull up figure 1, what happens, what's called the Thatcher effect, is when you take a face and you turn it upside down, a picture, okay, and you turn the eyes upside down and the mouth upside down, you can't really notice it. You notice there's something odd about it when the picture itself is upside down, but when the picture's right side up, it pops out at you. In fact, it's creepy looking. It was called the Thatcher effect, excuse me, by the way, because the first time it was ever done, it was done with pictures of Margaret Thatcher. So this works in humans, of course. And in fact, little babies even, when they look at a stimulus, like when they're 12 hours old, if you show them 3 dots, like 2 on the top and 1 on the bottom, they look at that longer than they look at something with, they can distinguish between that and something with 1 dot and then 2 below. So the idea there is they have a face recognition module built in. So there's something special about how we recognize faces. This is the first paper that's done this with any other primate. So what these guys did, what Rob and I think one of his postdocs and one of his students did, is they took unique pictures, so ones that these monkeys had never seen before, and they'd show them the pictures, and they stared longer at the ones that were, well, weird looking, that had the Thatcher effect put into them. So, I mean, what this shows is that there is some module, more than likely, in not only in us and in things like chimps, because it looks like it shows up in chimps, but even in something as distantly related as old world monkeys. So what we're getting here now, it looks like, is conservation of this face recognition module in monkeys. And the thing is, you see, if you do this same sort of thing with, say, pigeons, for example, which we in psychology always do stuff with pigeons, they don't discriminate between upside down and right side up faces. They will see upside down and right side up as the same thing right away. They have no problem with it. This shows then that we like looking face to face at each other, right? We primates, which may have something to do with what allowed us to become the sort of reciprocal altruists that we are. The fact that you scratch my back, I scratch yours, and I'm not just nice to my relatives. I'm also nice if Mark lends me $5 today, I'll lend him $5 tomorrow. The sort of, Hamilton and Axelrod reciprocal altruism thing. And to do that, you have to be able to recognize individuals. And you have to recognize their faces, of course, not just by, say, smell. And the other thing this fits really nicely with is some work that a guy named Dave Parrott did years ago, where he found cells in the occipital lobe of monkeys that actually respond only to pictures of individual monkeys. So when they see Eddie the monkey, that cell fires. But if it's Steve the monkey, that cell doesn't fire. So you put this all together, and it seems that for probably like 30 million years of evolution, perhaps, we've had this face recognition module, and this now, it's pretty clear that this shows up, and it works exactly the same way in these monkeys that are quite distantly related to us, as it does in you and me and everybody else here.
Let's, Burke, I sent the email, sorry to interject here. Sure. I sent Burke the paper, so go to page 2 if you can. There's a PDF. So you should be able to see it and pull it up on the screen. And what we're looking at is a picture of somebody in their late teens, maybe. It's hard to say. I really can't tell. They're upside down. It's pretty wild.
Yeah.
Okay.
And then you can see the effect if it's there. But anyway,
okay, go ahead, Vincent. You had a question?
So, Dave, I had a student once who couldn't tell data upside down and right side up. So maybe there are lesions in humans that cause similar things? Is that known that you can have a specific lesion in this area and cause a similar phenotype?
Upside down, right side up, but not faces, right?
Right.
Yeah, I mean that's the difference. I mean, there is a lesion that you can get that they'll get rid of your sense of left and right. And there's also the idea of contralateral neglect, where you ignore the left half or the right half of the world. That's the one thing that Kramer got when Crazy Joe Davola kicked him in the head. Yeah, you can see the picture now. And the one on the left, you can notice probably the one on the right there. It looks a little odd, but now I don't expect Burke to do this, but if everybody out there that's watching stands on their head right now, they'll notice that the picture that now is on the right, but it's upside down, it'll be on the left now. It's strikingly, there's something wrong with it, because what the researchers have done here is they've actually taken the pictures, the eyes, and flipped them upside down, and the mouth has been flipped upside down, but it doesn't pop out like it does. And
I don't see it at all. I don't see. So we're looking at 2 pictures of persons side by side. They look identical.
Yeah. But if you turn your head upside down, Mark, you'll see that it knocks you over. And the same thing happens with the monkeys.
Just do it with your laptop. It's quite impressive.
I mean, to me, it's really take the laptop and flip it around.
Wow, that's pretty wild.
You know, for me, because his lips are so strikingly colored differently than the rest of his face.
Yeah,
I pick up on the difference with the lips immediately, and with the eyes, it's much more subtle.
And in fact, with Margaret Thatcher, of course, when it was originally done, she has pretty thin lips, right? So it's actually much less noticeable in Margaret Thatcher. But
she
they
has
did it with
droopy eyes.
Well, she's got that. You got the Paul McCartney eyes, but they did it with actual monkeys. So they're showing monkeys pictures of Thatcherized monkeys, and the monkeys were noticing it. So I mean, it's working with them, and that's the amazing thing here.
Yeah, for sure. So I'm wondering, Dave. I mean, so I just remember being, in art class as a kid, and one of the techniques that they used to use to help us learn how to draw a picture of a person's face is they would give you a picture and they'd have you turn it upside down, and then draw what you see, and you were able to really do a much better job of making a drawing that really looked like the face that you were given. Does that connect in here somewhere, somehow?
Well, I think it actually makes a great deal of sense, because if we have this specialized face recognition cognitive module, we're probably going to find it very difficult to put imperfections in, to make the eyes a little bit too far apart, even though they maybe are supposed to be, to make the eyes a little bit droopy, to make the mouth asymmetrical. It's probably very difficult to do that if you're not an artist and you just have that kind of knack, because that sort of cognitive module is going to take over. So, and that would actually be a really cool experiment to do, he said, and putting that aside. There you go. Because I mean, I think in fact, I'll tell you, I might get a student on that next year. That sounds pretty cool. But yeah, that makes a great deal of sense to me.
Yeah.
So when did this, when do you think, can we tell by the type of monkey that this is being tested on, how far ago that this may have started? And how does it benefit us to have this?
Well, benefit us, I don't know. It's Rob, my friend Rob, he just finds stuff out because he can. But, well, I think it actually, in all honesty, though, it really could benefit us because we can figure out, think about things like when people do have brain lesions, when they have strokes, things like that. Perhaps training them to recognize faces is something that either we can learn a new way to do it, or realize that it's going to be very difficult to do, and you know, because people can be taught to do a lot of things after a stroke, for example. When did this happen? It looks like, if you look at this, considering the kind of monkeys they are, according to this paper, about 30 million years ago.
Wow.
Yeah. So it's what made us, this may be one of the things that makes us us, this ability to recognize faces.
Us and monkeys.
Yeah, well, exactly. So yeah, we don't throw our poo at each other, but beyond that, yeah.
Some of us don't. Okay, so we're...
So is it a primate-specific thing, or
seems to be, yeah. Because I know it doesn't show up. Doesn't seem to show up in other mammals that have been that people have tried. It's hard to find something nearly as visual as other primates, though, in mammals. I mean, that are commonly tested. Let's say that, like, you wouldn't use rats; they don't see that well. But I know it's been tried in birds, and birds are exceedingly visual. And birds, there's no difference. Upside down, right side up, sideways. They notice things right away because they're, I don't know, because they're birds or something.
But with birds, well, you know, aside from the fact that they're bird brains, right?
Yeah.
But with birds, they also see a different range of light, though. I mean, don't they see pretty much out into the UV?
Yes, they do.
And a lot of white light they don't see well.
That's right.
So
lot
they
of birds
may be
see
really
UV.
recognizing different things.
Yeah, and in fact, there are all kinds of data out there suggesting that birds recognize. That in fact, like some birds have, this going pretty far afield here, have an ultraviolet patch on their breast. So the say the male might have that and the female doesn't. So the females can recognize males by looking at something we can see in colors we can't even see, dude.
Blow your mind.
Well, that's pretty wild.
Yeah.
So 30 million years of behavioral qualities of the human brain. I mean, our human brain has been able to do this for 30 million years. And wow.
Pretty cool.
Yeah, it is pretty neat. I was wondering if we should go over to Vincent.
Yes.
And talk a little bit about the virus story. So, as mentioned,
this is a great story. This is a story about the discovery of an antiviral, and this is a beautiful paper. If you ever want to teach in a course how to get an antiviral drug, this is the paper to do. It's got everything. So the virus target here is hepatitis C virus. This is a human virus. It infects a lot of people. It's bad news. There are about 200 million people right now infected. It's spread by IV drug use and sex, or as my colleagues like to say, sex and drugs. And you're infected for life, and eventually you develop liver cancer. So it's not a good virus to have. So many companies are making antivirals for it. Now there's a treatment already now, but it stinks. If you get infected with Hep C, you get interferon, and about half the time it works, and the other half of the time you still have virus. Plus, the interferon has a lot of side effects, so it's not a good drug. It's not.
How many people actually follow through the interferon treatment? Isn't it like 50% can take it, and then after that?
That's another problem because it's a lousy treatment. You feel really bad, and so a lot of people stop, and that makes it worse. But it's far from ideal because it doesn't work most of the time. You want a drug that is nearly 100% efficacious. So there are 2 drugs in the pipeline, and they inhibit enzymes of the virus. So an enzyme is a very easy target to look for inhibitors. It's easy to set up an assay, and it's easy to validate. And there are 2 enzymes that this virus makes that it requires. One of them is a protease, and the other one is a RNA polymerase. That's an enzyme that duplicates the viral genome. Now, for HIV, we have antivirals against the protease of that virus and the polymerase. So this is a tried and tested route to get inhibitors of those. And as I said, there are drugs against those that are in the pipeline. They're in clinical trials. They're going to be used. But this company wanted to get something against a different target. The problem is the other targets can't be assayed. A lot of the other proteins are essential, but you can't assay for them in a typical antiviral screen. So what they did in this paper, this is called chemical genetics, by the way. This company, Bristol Myers Squibb, they have about 1 million compounds sitting on their shelves. So they took this whole 1 million compound library and screened through it to look for inhibitors of the virus. It's called a high throughput screen because it's done in multi-well plates, and it's actually done by robots. The robots put everything in the plate, they incubate them, and then they read the results. And then you get the results on your computer screen and you interpret them. So you can do 1 million compounds in a relatively short time. By contrast,
sometimes podcast hosts do it too.
Yes, and I was just going to say,
long days though.
So Mark, if I was thinking today, if you were really good, you could do 100 compounds a day, maybe. Would that be reasonable?
I could do about 10,000
a day. No, not high throughput, just by hand.
By hand. So as a graduate student in the Dave Thomas Lab at McGill, I would tip my boxes by hand, 5,000 tips. That'd take a couple of hours. Purify the enzyme. Load up the plates, load up the inhibitors, run the test. It would be done in a day, 5,000 a day on the bench. That's using hand pipe. You fill these little tip boxes. For every drug, you need a new tip. So I could do about 5,000 a day by hand.
You're good.
In 96 well plates, but if you
and that's a motivated graduate student.
Yeah,
that's very good. But to do
it's not a million.
Take you many days,
No
right?
way.
No, not close.
But anyway, we're all automated now, so they're able to go through 1 million. So what did they look for? They had a assay for whole virus replication, but it was a colorimetric readout or a luciferase readout. So it's an assay that can easily be measured by a robot. They screened this 1 million compound library, and they got a bunch of compounds that inhibited the virus multiplication. Okay, step 2. Sorry?
What was the target?
The target is simply virus multiplication. Okay?
Really? Just as simple as that?
It's a very broad target. So you're going to get multiple inhibitors. And this is where the next interesting step comes in. They don't want to get more inhibitors of the protease and the RNA polymerase because other companies already have those. So they rescreened all those positives that they got from the first round, and any drug that inhibited the polymerase or the protease, they threw out. Okay? So they did a separate screen.
So that was like a counter screen, right?
Sure.
Exactly. They want to throw away inhibitors of those two enzymes. And then what they had left, they narrowed down to one compound. Now, out of a million, one compound, the molecule is a very interesting, long, many-ring compound. It was very, very potent, but not at first. They got one compound that inhibited very well. And then they began to tweak it. They began to chemically modify it. And eventually they made a compound with incredible potency. The 50% inhibitory dose of this compound is in the picomolar range, like 5 picomolar. This is incredibly low. And they say that this is the most potent antiviral against HCV that's ever been developed. Right? So what's the target? It turns out that the target for this is a viral protein with no known activity, that at least that can be assayed. It's important for the virus. It's essential. If you take it away, the virus doesn't grow. But we don't know what it does. And we would never be able to set up an antiviral screen for this protein specifically. The only way we could get it was to do this negative or chemical genetics approach. So the protein's name is NS5A. Yes, Mark?
Oh, I was just wondering, there's only, how many proteins does this virus make?
Yeah, maybe a dozen at the most.
Yeah.
And so there's not a lot. But this one was a target. And so NS5A, and they know that the drug is hitting this because the drug binds to that protein. And in fact, they do get resistance eventually with this compound. They get viral resistance, and the resistance mutations map to this protein.
It's amazing.
But
I think
the neat thing is
I just think that one of the distinctions here is that, frequently when, people in big pharma and biotech companies do these types of high throughput screens, they tend to be very specific for a given protein target,
right.
Whether it be some sort of membrane receptor or some specific enzyme. And the key here with these guys is, if I understand correctly, is that their assay, their initial high throughput screen assay, was looking at viral replication. So instead of just honing in on a single protein, single gene product, or a couple of gene products, they said, let's just look at the whole thing. And so then they were able to really pull out something for which we have no understanding of its function, other than now we know for sure it's required for the virus to be replicated.
I see a couple of issues, though. One issue is that they couldn't replicate the virus in vitro for a long, long time, right?
Right. So this is only made possible recently, although there are ways to get around that. You can use what are called replicons. These are very small RNAs which, when put into cells, will replicate. You bypass a lot of the steps. So as George was saying, if you screen, you're going to miss a lot of the steps that could be inhibited. But yes, the ability to grow the virus very recently was key to all of this. But even more so, the fact that you could eliminate the 2 known drug targets, the only ones that are enzymes, as far as we know, was really important for this, because it allowed you to say, I have something that inhibits what, beyond the polymerase and the protease, what is it targeting? And that's why they are able to get this target, which doesn't have any enzymatic function. So it turns out to be very potent. They did a number of studies. This paper has everything. So they discovered the drug, they refined it to make it very potent. They put it in animals to show that it was safe, non-toxic, and then they give it to people initially to see if it's properly absorbed, and it is, and if it goes to the liver, because that's where the antiviral needs to go for this virus in order to be efficacious. So it gets to the liver very effectively. And then they look to see in people with hepatitis C what this did to their virus levels in the blood. And they try typically 4 or 5 different concentrations, and they find that one particular concentration, 100 milligrams, which is a perfectly reasonable amount to be taking of a drug, really knocks down viral replication significantly. So this is an extremely...
How long did it take him to do all that?
That's a good question. I mean, there are a lot of authors on this paper, and so I would suspect it took at least a year to do this, which...
is amazing. Usually the medicinal chemistry will take a year. Getting down to picomolar, they really, I guess they precluded a little bit of the medicinal chemistry by screening a large library. They're looking for things that already started.
They only had this one hit, and they, then they modified it substantially. They say they're going to publish that separately. They say it's going to be a series of separate publications. So presumably that's very interesting as well. But in the end, now you have a drug which has already been in people, so it's going to be trialed more extensively, and so this could be very promising. The only drawback, as one of my colleagues has said, the fact that they're publishing it means that they're probably not going to sell it, and they have something better already.
Yeah, you're not supposed to publish this kind of stuff. You only throw out stuff, but it depends on the competitive environment, right?
Yeah. Well, you could quick search in the patent database. You'll see if they patented it.
Well,
I'm sure they have.
So there's no.
Right? If they published it, their lawyers would have told them. But my colleague had been in a pharmaceutical company for 30 years, and he said you never publish unless you have something better. So I don't know if that's true.
You're ready to go. So here, if you publish it, you have to have that patented because you're basically throwing the photocopy into the ocean. So, but every year, so with a drug that could save potentially 200 million people. I'm just talking about the economics of it because this is important. The only way to drive innovation is to know where the money is. And so you have 200 million people. That's potentially $1 billion market per year, roughly. And for every year that you're not ready to sell it on the market, so you have to do your clinical trials, but you're already patented, your patent is running out, so you're losing $1 billion a year, right? So chances are they've derived the structure beyond the current patent. They probably patent this anyway. They patent everything. They have to. And just in case somebody leaks, I mean, there's so many authors. And then they've got something that's outside the realm of this patent that maybe they haven't patented yet. Who knows? That could extend the life of the patent. But that's a whole. I think what's incredible here is they have a picomolar EC50 in the picomolar range. That's nothing. That's like a flake, a crystal. The drug.
It's amazing.
You put it under your tongue, and the virus gone.
Yeah, and in the human trials, the efficacy is similar. It's very, very low. And one more thing I want to point out: they go on to check combinations of this drug with the other two drugs that are in the pipeline, and those combinations are synergistic. In other words, the effects are not just additive; they're more than additive. So, using multiple combinations of drugs is very good for viruses because they can easily mutate to become resistant. But if you use 3 drugs, the probability of mutation is very low. So a fantastic paper, hopefully a great drug to treat people who need it. But in terms of teaching, this is just fabulous because everything is right here.
It's a real tour de force.
How does this compare to an HIV drug where HIV, you have to stay on it because doesn't HIV integrate itself into your genome, so you have to constantly for the rest of your life suppress it, versus HCV that if you clear it.
Yeah. No, so HCV does not integrate it like HIV does. So if you can reduce virus replication, you can clear virus completely from the individual. And in fact, some of the people who get interferon, in fact, are cleared and they're healthy and they never have infection again. So this also has the potential to do that.
Well, it's a pretty amazing story. And one, they've done what pharma don't normally do. Pharma will follow the protease, polymerase line of development because that's something they know how to do. They usually buy up a company that's developed something novel. In this case, I guess they're the pipeline for their current drugs, or maybe they're coming off patent soon, and they need something radical. So they made a radical step and screened using cell-based assay, which is it's hard to tame these cells, right? There's so much variation that can happen when you're working with living. It's like working with doing film with kids and animals, right? Cell-based assays. That's pretty wild. One of these, a paper like this comes out once every couple of years. It's not every week.
Absolutely. This is an amazing paper. I haven't seen something like this with respect to antivirals in a long time. This is better than many of the HIV antivirals because it's a beautiful linear sequence story and it's so potent. And it's a target that you could not search for any other way. So that's why it's beautiful.
And it's a new approach. Maybe there are some viral proteins that we haven't targeted yet that are brilliant targets.
Sure.
Right? It opens up the door for what can become a drug target. And so that would, expand not only across viruses, but bacteria as well. And it's pretty cool. Well, it's very cool.
Yeah.
A question from the chat room. What's the molecular weight of the antiviral?
What a good question. In fact, it's a little more than 700 Daltons, which is kind of big, but nevertheless, it's very well absorbed, they point out. So kind of large in terms of something you'd take orally, but nevertheless very bioavailable, as we say.
Right.
The other thing I was thinking about with respect to this particular study is to basically get yourself to the point where you really only have one pony in the stable, just the one hit after the counter screen, and to be able to take that all the way through, an SAR study, a structure activity relationship study, and get that potency so incredibly high and be able to basically bring something to market, having just one pony in the stable. That's impressive.
And yeah, not to mention the 200 million lives that they're going to save potentially, which is, ironic. It's just incredible. It's
including Pamela Anderson. I'm sure she'll thank them thoroughly.
I probably think that she's cleared her virus because she's still doing fine. It does go away. It clears, but not. 50% of the people, I think it is, something like that order of magnitude, don't clear it and then have to go undergo liver transplant. And then they reinfect their new liver as well, and they develop cancer. It's pretty harsh disease. But, good news. That's very, very good news as well. Not only is it cool, it's very good news. So I'm going to take a minute to thank our sponsors. Today, this episode is brought to you by Audible.com. Audible is a leading provider of audiobooks with more than 75,000 downloadable titles across all types of literature, including fiction, nonfiction, and periodicals. So for the listeners of Futures in Biotech, Audible is offering a free audiobook to give you a chance to try out their service. Today's pick for the week is the Genome War, how Craig Venter tried to capture the code of life, in part because he had a major story that came out yesterday that's potentially huge, maybe not. And sort of this book kind of reflects some of his entrepreneurial approach towards the life sciences and towards bringing innovation. Right? He was he was the geneticist that brought the cDNA library. That's a tool in genetics. It's not the tool, but it's a very, very good tool that allows you to clone genes, especially over the 80s and 90s. It allowed us to clone most of the genes that were cloned during that era. And but his point of doing it was to actually get patents on every single gene, which is an important ethical question. Should we patent genes and have exclusive rights to developing targets towards those the proteins that they make? Anyhow, it's fairly controversial. So you can read The Genome War, how Craig Venter tried to capture the code of life. And now, so that he doesn't preclude me from any interviews, because I might want to try and get him on the show. We'll also suggest potentially his book, his autobiography, A Life Decoded, My Genome, My Life. There's a good counterpoint to some of the arguments made in the first book. So if you want to download...
Is he the one reading it?
No, it's by Dick Hill. So the first book, The Genome War, How Craig Venter Tried to Capture the Code of Life, is by James Shreve and narrated by Grover Gardner. And A Decoded Life, My Genome, My Life is, of course, Craig Venter, and narrated by Dick Hill. I don't know. Does he have a good voice? probably one yelling at people. He's kind of like the Steve Jobs of biotech.
He gives a good talk.
Yeah. He's but he owns the human genome. Did you know that?
He is the human genome. He's got the reference sequence.
Yeah,
he is.
He owns his genome.
Right. So you can download the book for free by heading over to audible.com/biotech. Sign up for 14-day free trial, and the download is free. It's a win-win situation, because if you decide you like Audible, you can stay with them. But if you don't, you can keep the free audiobook. So it's a free audiobook for you guys. I download iWoz myself when I did this. That's audible.com/biotech. We really thank them for the support of Futures in Biotech. So let's continue. Let's I don't know. Should we go hardcore DNA and then go? Let's do that. How about George? Okay. How about you present your paper, because it's pretty wild. And for the audience out there, hang on to your hats, because this is some pretty hardcore
molecular biology.
Yeah,
protein engineering. So what I'd like to review with you guys today is an article entitled "Encoding Multiple Unnatural Amino Acids via Evolution of a Quadruplet Decoding Ribosome." Ooh, fancy. It came out in the March 18th issue of Nature, and it's basically the latest installment from Jason Chin, who works at the MRC in Cambridge, UK, and it's basically these. Jason Chin, a bunch of years ago, was working with Pete Schultz, and Pete Schultz is at the Scripps Research Institute. And what Pete's been interested in doing for many, many years is basically synthetic biology. So the idea is you have a number of proteins for which these proteins interact with each other. And to look at those interactions, typically what a biochemist might do is use a cross-linking technique whereby you throw in a chemical and you link these proteins to each other to see which of the proteins in your mixture actually come into contact with each other, for instance.
So two proteins floating by each other. Yep. You have a chemical that says, I want to attach both.
And the problem with that is that frequently these sort of chemical cross-linkers are relatively non-specific. And frequently they'll really alter the function of the proteins. So Peter Schultz and a number of others, probably about 20 years ago, started to try to basically incorporate unnatural amino acids right into the protein. And these unnatural amino acids would include the chemical crosslinker. So your chemical crosslinker, instead of being introduced outside of the protein, would be actually included in the protein. So the way that they did this was they took advantage of a little genetic trick. It turns out that in E. coli, in bacteria, there's an amber codon. The amber codon is a codon that basically in the messenger RNA that codes for a translated protein, it gives a stop signal. It tells the ribosome when to stop synthesizing, when to stop interpreting, reading the messenger RNA, to stop reading it and release the protein into the cytosol of the cell. And it turns out that there are some suppressor mutations whereby instead of that stop codon giving a signal to stop translation, the protein reads through, or the translation machinery reads through that stop codon. And these are called these amber suppressor mutations. And so what Peter Schultz did was he took advantage of that a bunch of years ago to put in the tRNA that would basically recognize that, or would represent that stop codon, or be recognized by the ribosome, pair it up for that stop codon. And a tRNA that would have one of these unnatural amino acids attached to it. And sure enough, he was able to incorporate all sorts of interesting cross-linkers into proteins, and he was able to use this to really pinpoint cross-linking of various proteins to each other. But it was relatively limited. And first of all, in terms of efficiency, it turns out that frequently incorporation of your unnatural amino acid is very small. It's down below 10%. So 10% of the protein that you synthesize actually has the unnatural amino acid incorporated into it. That's the first sort of big limitation. And then the other limitation is that you can only do one at a time. And so that's potentially a little bit of a problem. So what Peter Schultz did when Jason Chin came to his lab is he's challenged Jason to basically take a genetic approach. And a bunch of work that Jason did in his lab, and I'm trying to summarize a lot of years of work and get you to the real interesting
Let me ask the question.
Go ahead.
Yeah. So what you have, right, is a protein making machinery in your cells.
The ribosome
zips along RNA, because RNA is the message, right? You kill the messenger. So DNA is the code, RNA is the messenger. So you zip along the messenger RNA and you translate that into protein. And he was working on mutations of the ribosome, the zipper part, right? Not the zipper itself, but the zipper latch that attaches the amino acids. And he screwed around with it until it started to make mistakes that he wanted, right? But it was limited.
Very limited. Limited in the sense, well, they took advantage of things that people had seen in the past, basically along the lines of what you just described. Basically, it would make a mistake. It would skip over a codon that would tell the ribosome to stop synthesizing the protein.
Okay, and then that's where the mistakes would happen.
That's where the mistakes would happen. And what he did was he introduced a tRNA, which brings in the amino acid for synthesis on the ribosome, that included his unnatural amino acid, an amino acid that contained some sort of chemical, like a chemical crosslinker, that he'd like to use to study the function of the protein. So the limitation was how efficiently was this unnatural amino acid incorporated into the protein, and the fact that you could only do one, maybe two of these unnatural amino acids at a time. But they wanted to go much further. They wanted to expand it to be able to put in any sort of unnatural amino acid that they wanted, anywhere, anytime, and multiple locations. So in order to do this, you've got a couple of really big hurdles. And the biggest hurdle, of course, is you have this endogenous bacterial ribosome that's trying to translate all of the proteins that the bacteria needs in order to survive. And if you start throwing in some bunch of crazy unnatural amino acids, you're going to totally scramble all of the proteins inside of the cell. It's just ridiculous. So Jason is very clever. The first thing he did was he said, let's engineer a ribosome that would be able to recognize a very specific messenger RNA, but not any of the other messenger RNAs inside of the cell. And he was able to do this by taking advantage of the fact that in the messenger RNA, there's this little sequence called the Shine-Dalgarno sequence, which matches up with a little bit of sequence inside of the ribosome and is part of basically getting the messenger RNA docked into the ribosome.
So let me see if I can get this.
Go ahead.
I was like,
go ahead. Yep.
My dad's listening, so I want to be able to explain it. So you have a machinery that works.
Yep.
Now you engineer machinery that works completely differently.
Yes.
It doesn't recognize the RNA from the human cell. It recognizes
from the bacteria. Yep.
From the bacteria.
Yep.
It recognizes foreign RNAs or different types of RNA.
Completely heterologous RNA.
Okay, so this is RNA, a message from someone else.
Yep.
Okay, and here's what's
what's going to happen inside of this bacteria, right, is you're going to have two different types of ribosomes. You're going to have the natural bacterial ribosome in there, translating all of the proteins to keep the bacteria alive, And in addition to that, you're going to have this, you can call it an unnatural ribosome. Jason likes to call it orthogonal ribosome.
Sure. Sorry, this is going to be for better audio.
Sorry, guys.
Yeah, that's better. All right.
So you're going to have two ribosomes, two separate ribosomes that exist inside of this bacteria. One's the normal wild type ribosome that's going to translate all of the machinery that's necessary to keep this bacteria alive.
Sure.
And then the second ribosome is going to be something that's not going to interact with the normal messenger RNAs in the cell, but is only going to be able to recognize messenger RNAs that you program into the cell for expression on your orthogonal ribosome.
So you're not going to modify the life of the organism, and you'll have a completely discrete engineering set of tools inside that cell.
Exactly. And that's exactly where they're headed with this, right? Is they want to basically generate essentially a protein toolkit where you can go in and you can monkey around with your favorite protein and do basically whatever you want to it.
Alien.
The implications are huge. So Jason took it one step further. He said, okay, now wait a second. If I synthesize my protein on this orthogonal ribosome, I'm going to want to be able to incorporate normal amino acids, and I'm also going to want to be able to incorporate these unnatural amino acids and multiple unnatural amino acids. So if you think about how the codons work that specify these individual amino acids, it's 3 bases at a time.
Right.
That limits the number of combinations. And it turns out that if you take all of the possible combinations of 3 bases, everything's spoken for in terms of the 20 different natural amino acids that we have.
Yeah, but that's like 4 billion years of evolution saying 3 codes, 3 bases in the DNA are fine.
Yep.
Okay, so he's saying...
So what he said was, instead of having 3 bases for the codons, we're going to have 4.
Uh-oh. Okay, so when you land on Mars and you're sequencing DNA, right, that's kind of DNA you'd find if there was DNA. This is completely alien.
Yes.
There's nothing living about this. This is completely but it's
alien.
Alive. It's an alive synthetic.
So what he did was he took his orthogonal ribosome and he started to make mutations in the decoding zone. This is the zone on the ribosome where these tRNAs come in with their amino acids and they dock in and they match up with the messenger RNA that's specifying the sequence of amino acids that are going to go into this protein. So that decoding zone in the ribosome, he started to mutate it. Mm-hmm. And what he did was he took advantage of a selection process. And the selection process was chloramphenicol resistance and antibiotic resistance. So he put in his unnatural messenger RNA, a messenger RNA that would not be recognized by the wild type and the endogenous ribosome of the bacteria. He put into that messenger RNA was a gene that would basically make the bacteria resistant to chloramphenicol. Mm-hmm. But one of the amino acid codons would be one of these 4 codon sets. And he then selected in the lab, he did evolution in the lab of the bacteria to generate a ribosome that would be capable of recognizing quadruplet codons.
4 is better than 3, right?
4 is better than 3. Why is 4 better than 3? Because now, instead of being limited to 20 amino acids, you have something like 200 possibilities of different types of unnatural amino acids you can put into this thing.
Oh boy. Do I have to learn the genetic code again, George?
No, you don't. I'm sorry.
Can I ask a question, George?
Yes, please.
So he selected on a antibiotic. So if you could not adapt to a 4 codon decoding mechanism, you wouldn't grow on the antibiotic. Do I understand that correctly?
That's correct.
So what was the frequency? How many steps did he have to select in order to get that to happen?
So I think the frequency with respect to, how many, so he did random mutagenesis, and then I think he put the DNA into roughly 10 to the 9 cells, and I think he had a total of 14 hits, and they were all different sets of mutations.
And each one of those was sufficient to allow the ribosome to decode 4 bases at a time.
Yes. No, I'm sorry. It wasn't 14, it was 3. So 3 out of 10 to the 9 cells.
So do we have any idea what this ribosome looks like? It must not be very different from normal ribosomes, right?
Yeah, so what appears to be the case is that decoding portion where the tRNA comes in and brings in the amino acid with it is enlarged to accommodate, presumably, the larger codon.
Yeah, maybe that means that at one point we had such a ribosome because the ease of getting it to recognize 4 would suggest that, no?
Yeah, it's a distinct possibility. I agree. But I think a lot about the implications of being able to make these types of changes to proteins. And, there's a big field right now where people have basically been re-engineering proteins, enzymes, to get them to carry out very specific chemical reactions. And now basically these people are going to be able to look at the active site of that enzyme. And, let's say there's some very specific chemical reaction that's useful for some industrial process. They're going to be able to come in there with unnatural amino acids that put in the specific functional groups necessary to carry out all sorts of crazy different types of chemical reactions and basically generate catalysts, biological catalysts, to carry out all of these chemical reactions that we're now, expending huge amounts of energy and time the old-fashioned way.
I think there's 2 things that are amazing here. One is changing the genetic code to a 4 codon language, right? Allowing for a huge amount of synthetic biology that was never imagined before. You could now create therapeutics that are protein-based, for example, maybe perhaps an insulin that has chemistry that you never had before. And it's just, I think it's hard to really translate the importance of this paper to the general audience, because, okay, so they changed the genetic code, added a base. Well, this hasn't been done in 4 billion years, right? And this is complete human synthetic construction of and manipulation of life in a way that's never been done.
And sometimes when you see these articles on the face of them, you say to yourself, oh, well, did they do that just because they could? And the answer here is they have their eye on a particular prize, and that is this protein toolkit. The
I thought
ability
it was
to
total
do real
world
synthetic.
domination. You disappoint me.
Well, yes, and I welcome our new 4-base DNA organisms overlords.
Exactly.
How we said that one before?
I think I said that.
Our FIP hosts are always welcoming our new overlords. So
that's right.
It's a crazy story. I'm glad you brought it because I would have read this and I would have looked at the title, encoding multiple unnatural amino acids via evolution of quadruplet decoding ribosome.
So look, I mean, for the biotech people out there, right? You know, Vincent was talking a little while ago about the high throughput screen. You know, a lot of times these high throughput screens are fluorescence based, and they look at protein-protein interactions. And you have a fluorophore of some sort on one protein and a different fluorophore on a second protein. And when the two proteins interact with each other, you get sort of a synergy in the signal. And there's a change. And sometimes these assays that people put into these high throughput screens are on the basis of looking at changes in whether or not that signal is increased or decreased. This technique basically says to you, well, gosh, you know, we can make an unnatural amino acid with whatever fluorophore you want and put it into pretty much so any location in any protein that you want. It's a new tool that I think will lead to the development of better, faster, high throughput types of screens where for people out in big farm or even in biotech looking to look at protein types of interactions or things that alter the conformation of the protein.
There's a billion things you can do. I think if you look at
it's a tool.
Some of the chemistry you're limited. You know, chemistry, you're adding molecule here. You're taking a molecule, adding an atom, removing an atom, doing a few chemical reactions. You can synthesize things that are fairly large and complex, but you can't synthesize anything chemically that looks like a protein complex. Right? It's just impossible. It would take you your entire life. So recruiting life and then really, this is the largest manipulation of genetics I've ever seen. So we'll have to get Jason Chin on the show. And maybe he could explain some of the reasons why he built this machine.
Yeah, probably better than me.
Well, no, that was really, really good. I'm convinced now. I'm convinced. And I don't tell Craig Benner because he'll own it too. The 4-base code. He'll patent it. No, I hope this is, well, we'll have to find out. Maybe we can patent it or get license it. So thanks, George. Let's move on to Andre's story. Andre has a pretty wild story. We're having some microphone banging. Is that Dave?
It's not
okay.
me.
So if you're holding your mic, hold it tight. SM58s and PG58s have to be held tight, or else it makes noise. Okay, so Andre, you have one of the wildest stories,
yes.
and it's somewhat scary as well. Why don't you go ahead?
Well, so essentially my story is on the publication of the Neanderthal genome, and when you made this suggestion that I should cover this, I said, oh yeah, sure, I'll do that. So this is essentially my lesson for the week, is that the next time I tell Mark I'm going to cover a paper for FIB, make sure to check the paper first. I mean, this thing is probably one of the largest, most complex paper I've ever presented. I mean, I'm going like, oh, it's in Science. It'll be 2 pages, right? 56 figures, 63 tables, 53 co-authors. I mean, I work in genomics. I mean, it's a field that's well known for its monster paper, but that one was just huge.
By the way, if anybody wants to, they can go back and listen to one of the early episodes of FIB where I had Svante Pabo and Jason. Well, no, not Jason. It was Tom from 454. I can't remember his last name. He's the one who says in the theme of the show, he's the last quote. So you can go back and listen to some of the rationale, how they were doing it. It was a mid project that Svante discussed. And he called me a chimpanzee, so it's kind of interesting, kind of fun. He has a great sense of humor. But not to interrupt you here, Andre, go ahead.
No, that's actually that's what I was about to say. That the team was led by Svante Pabo, who's a geneticist at Germany's Max Planck Institute for Evolutionary Anthropology. And so essentially what they're doing is they want to study the Neanderthal genome. And before I go on to the import of this story, we have to do a little paleontology introduction as to who were the Neanderthals, Because, okay, they may be the butt of a bunch of jokes, but it turns out that Neanderthal man is not an ancestor to modern man. They're actually a separate lineage that's separated from ours about 400,000 years ago. And I love the words I read. They wandered out of Africa, doom-ba-doom-ba-doom-ba-doom, and boom, they're in Europe and Central Asia. In the middle of the Ice Age, conditions for which they were perfectly adapted. And during that time, essentially climate change had very strong impact on the patterns of human evolution. And during the Ice Age, not only did you have glaciers coming down, there was also desertification in Central Africa, which essentially prevented modern man from having contact with the Neanderthals for several 100,000 years. And then the climate warmed up a little bit again, so modern man could go up again through the Middle East. And they were essentially in contact with the Neanderthals for between 30 and 50,000 years until the Neanderthals disappeared about 37,000 years ago. And people tend to think of that as like tribes of Homo sapiens invading Europe, driving out the bastard Neanderthals out to the sea. And you have to realize the enormous length of times that for this to take place. We're talking about 30 to 50,000 year cohabitation between those two human species. So it's like 1,500 to 2,000 generations. And just for fun, I said, well, let's say I'm a Homo sapiens tribe starting in the Middle East. I just got out of Africa and I decide I'm going to conquer Neanderthal land. So I'm going to have to walk towards Europe until I can push them out to sea in southern Spain. Well, it took 50,000 years for them to do that. So essentially, every year you just need to advance by 100 meters. And you do that for 50,000 years until you reach Spain. So this conquest of the Neanderthals by Homo sapiens took a very, very, very long time. And essentially, the crux of the paper was, and that's the reason it made all the newspapers and all the front pages, is that they're showing evidence that there may have been some inbreeding between the advancing Homo sapiens and the population of Neanderthals. So how did they... Oh, crossbreeding. Yes, I said inbreeding. Bad word.
That's British royalty. Sorry. I'm a subject of the...
So essentially, how did they figure that out? I mean, I've talked a little bit in an earlier fib where I talked about the mammoth genome, about the massive challenge of doing genomic archaeology, dealing with very, very old DNA sample that have been essentially standing around outside of a refrigerator for tens of thousands of years. I mean, you know how your lunch looks after a week. Imagine after 40,000 years what happens to the DNA. So usually they get it out of bone, which allows it to be a little bit protected. But invariably, the DNA is going to be degraded. It's going to be chopped into little pieces, and in this case, pieces of less than 200 base pairs. So that's tiny, considering you want to look at a 6 billion base pair genome.
By the way, Andre, you know how they practiced?
No.
On salami from New York. I swear to God, it was 70 bases. It was degraded to 70 bases, and they did bovine genomics beef.
Does it have to be from New York? Is that a requirement?
That's what they did. That's what they did. It's an inside story from 454 that we got from Tom over at 454. Go ahead. Sorry, Andre. They practice
well,
on
at least they found out the salami was made out of beef. Imagine with their surprise, they thought it would have been made of people. But anyway.
Wait a second. This is New York we're talking about.
Yeah, not Cleveland.
And it's made of chuds.
Chuds. Nice.
That's some good salami.
That's a very old sci-fi reference. Okay.
Well, no, across a mile from my house in Shaker Heights in Cleveland, across the tracks, there was a sausage factory, and there was a mass murderer just a few months ago. They keep finding new bodies. I think there was 18 bodies. Yeah. And so it was a sausage factory.
Good sausage.
Yeah, so maybe people wondered what the smell was, but...
Wow, well...
Okay, we distracted. Rat hole.
Yeah, exactly. You have the rat hole song. Okay, where was I? Okay, what happens to DNA after tens and thousands of years without segueing into mass murderers? The other thing that happens to DNA molecules, they become chemically modified in a very annoying way. One of which is that many of the cytosine residues are deaminated into thymine. So essentially you're getting C to T and G to A mutations that were not originally in the DNA. They come there because of chemical degradation. Another problem is that the amount of Neanderthal DNA is tiny. I read the term DNA swamping. So the amount of DNA from Neanderthal is like maybe less than 1% of what they originally isolate, most of which is coming from microbial organisms that will colonize the specimen after death. I mean, they are decomposed after all. Mm-hmm. And because of the tiny amount of Neanderthal DNA, plus the fact that, and that's what they found out later, is their DNA is very, very similar to ours. So any contamination from the people who handle the bones, handle the DNA before the sequencing, small amount of contaminations from these modern humans will affect the quality of the data. So the method section of that paper reads like a molecular obstacle course with tons of different controls and a way to account for any different possibilities. So they started from bones from 3 female individuals that were found in the Vindija cave in Croatia. Love Croatia, beautiful beaches. 2 of the individuals appear to be maternally related. They have similar mitochondrial DNA, and they were carbon dated about 38,000 years. So that's very old bone. And there's also a 3rd individual that's even older, but there was no collagen, so they couldn't do a carbon dating directly. So they actually used a sterile dentist drill to drill into the bone, make some bone powder, about 50 to 100 milligrams, in a clean room facility in Leipzig. And before leaving the clean room, they actually tagged all the small DNA fragment with a small molecular tag like TGAC at the 3' end. That way they know that all of the DNA that they see has to have that tag. If not, it was contamination coming in from later.
Sure.
They also enriched the mammalian DNA by digesting with restriction enzymes that would recognize site rich in the dinucleotide CpG. So that's more common in bacterial genomes. So that gave him like a 4 to 10-fold enrichment. There's actually a second paper in the same issue of Science where they use a million probe microarray to purify human-specific sequences. And that one's a much smaller paper. I should have presented that one, but that's another way of getting Neanderthal DNA for sequencing. And then
to wash away the human DNA.
Yeah, essentially. Well, they washed away any sequence that looked like human and chimp to try to get the human-specific sequences, and then use that to to get Neanderthal-specific DNA. Mm-hmm. But that's the second paper. So essentially, they use a 454 sequencer. They got lots of sequence. 95 to 99% of it was crap. They essentially compared it to lots of sequences from human, chimps, rhesus, mouse genomes, everything that GenBank, and anything that did not look like a mammalian sequence. And usually you can tell from the ratios of GC and the sequence similarity to other sequences. They were able to get rid of most of sequences that were of non-primate origins. And then they did a bunch of statistical tests to try to estimate levels of external contamination, looking at, for example, well, they were all female, so any Y chromosome sequences, they obviously came from contamination. They looked at mitochondrial DNA contamination. They actually did a pretty good job. The contamination was estimated at between 0.7 and 1.5%, which is really excellent. So what they ended up is essentially about 60% coverage at 1.3x coverage. So essentially, on average, every gene was sequenced 1.3 times, and they're covering only 6 genes out of 10. And as far as genome coverage is concerned, that is pitiful. That is really, really low coverage, bad quality. But hey, it's 40,000 years old. What do you expect? They compared that sequence to a variety of genomic sequences of human, and they were very broad in that. Initially, they did comparison from 5 different genomes and 3 non-Africans, so from Papua New Guinea, China, and France. I don't know why. Maybe that's where the graduate students came from. And 2 from Southern Africa and West Africa that they sequenced themselves. That's additional human genome information that they use for this project. And what they've noticed is that the divergence of the Neanderthal genome to a human reference genome, so that's the one that's already in GenBank, is greater than any of the individual human genome that they sequence. So that's good. They also estimated that they diverged 270 to 400,000 years ago, which is consistent with the fossil data. So that part, nothing new. Where things got interesting is now they started to look for genes and regions that, for example, are common in human and Neanderthals, but different from the chimps, our closest primate ancestor. And these are essentially, let's call them human-specific genes. And that's about 87% of the changes that they found. What's more interesting are the genes that are, for example, only found in modern humans, and Neanderthals and the chimps have the other version. So these would be essentially genes that have evolved later to produce modern humans. And there are actually very few of these mutations. Actually, there's a lot of these mutations in between genes and promoters, but if you look at the ones that change amino acid sequences, you're talking about 87 genes, only 5 of which have at least 2 amino acid mutations within the same genes. And you look at what these genes code, what's fascinating is, for example, so they had like 5, and there's another one that had an indel in it. So there's 6 genes that are especially interesting. 3 of those essentially affect skin morphology, which is kind of weird. So either we have soft skin and they don't. But one of them is involved in melanocyte pigmentation. Another one is expressed at high level in exocrine sweat glands and the inner sheath of hair roots. So maybe they were hairy and we were not. It's hard to tell what these genes' functions truly are just by looking at their function. One is involved in wound healing, for example.
We have to put them into human stem cells.
Yep. Well, they're gonna. Well, I'm guessing they're gonna mess up. I'm guessing they're gonna mess up the mouse humans first.
Do it.
I'm being facetious here. We could bring them back. We could bring the Neanderthal back.
No.
But go ahead, Andre.
What's also fascinating is they look for groups of mutations that have been what they call positively selected in the evolution of modern humans. So instead of looking at just like one little region, they look at larger groups of DNA where there's at least like 50 mutations that seem to have been selected for as a group. And then they look at the genes in these regions. And a lot of them seem to be involved in brain development. One of them is called NRG3. It's associated with schizophrenia. Another one called CADPS2 is associated with autism. There's another gene involved in cancer, which seems to be may affect aspects of energy metabolism in humans, considering that Neanderthals was adapted to cold climates, and while we're coming out of Africa, that there might be something in there. There's also another gene that lies in the Down syndrome critical region. Again, another gene associated with cognitive development. So these genes are like really, really fascinating in what they can tell us about what makes us human compared to Neanderthals or even chimps. But they're not the reason why this paper made the front page of almost every newspaper in the world. It's because they also broached the subject of kinky interspecies sex.
It's that whole sex and drugs and rock and roll thing again, right?
Oh, yeah. Well, rock and rolls being rocks, no rolls. Yeah, they haven't invented rolls. They had invented rocks, though. So the way they found that out is because they had access to human genomic information from a variety of human populations. You had Africans, which is essentially where the human species originated from, which then migrated into Europe and then on into Asia. So whenever they would compare Neanderthal sequences to a human population, they would always look more like the Europeans or the East Asian population than the African populations, which theoretically did not migrate out of Africa and mate with Neanderthals. The amount of Neanderthal sequences in the non-African genomes is pretty low. We're talking between 1% and 4%. And it was kind of very difficult to say this gene is Neanderthal and this one is not. It was mostly small SNPs, small one nucleotides changes. Yeah, exactly. But it is notable that the Neanderthal are as closely related to a Chinese or a Papuan individual as to a French individual. So whatever gene mixes took place, that occurred before these populations separated between Europe and Asia. And they also noticed that most of the gene transfer were between the modern human, the Neanderthal into modern human, although that could be simply because the Neanderthal samples that they all came from the same place, and they had a very small number of individual. There are some caveats out of this whole story. One is, and they address this caveat, they don't explain why, and I find that fascinating, is that this was not noted when they looked at the mitochondrial DNA genomes of Neanderthals and modern humans. It's much easier to look at mitochondrial DNA. It's smaller. There's multiple copies in each one of our cells, and it is maternally transmitted. You get essentially the mitochondrial DNA from your mother, and you don't get it from your father because sperm doesn't have mitochondria. And when you essentially do the same analysis with mitochondrial DNA from Neanderthals and modern humans, you do not see this. And I mean, it's getting me to think. I mean, maybe it's telling us something about how this gene exchange actually took place. There have been studies of what happens when a population moves into the environment of another population. We've seen this in America, and they've kind of realized that it doesn't take too many breeding events to create kind of a wavefront of genes from the resident population into the colonizing population, especially since the colonizing population is initially very small, and then the ones that move into the new territory have a very large contribution to the future population that ends up being there. But
maybe it has something to do with the way the breeding happened. Maybe it was male to female, male human, female.
That's what I can't help myself from thinking. I mean, one doesn't have to look at history very far to realize that a lot of these, let's call them breeding events, just to be nice, were not necessarily the result of a lifetime loving relationship. And they might be different effect whether one population would accept a mixed breed more than one other. For example, maybe they would, coming from a female Homo sapiens, it'd be, I mean, maternity is a certainty, but paternity is an act of faith. And if the offspring looked enough like a Homo sapiens, they're maybe more likely to be accepted. While in Neanderthals, maybe they would notice right away and kill or eat them. There's really no way for us to know how exactly that happened. It's going to take a lot more studies, sequencing other samples from the Neanderthals from other sites. And of course, as a cost of doing sequencing, it's going to tell us a lot more. And also the goal right now is to really increase the quality of the sequencing coverage. 1.3x coverage for a genome analysis is very, very bad. And they had to do a lot of statistical hoops to go around the chemical modifications, sequencing errors, and all of these things. So, I mean, this is really a very preliminary study, and I'm sure people are going to go over this as much as they can, looking at other populations, other sites where Neanderthals were found. And I mean, once they've done Neanderthals, they can start doing other humans as well, including Homo sapiens samples, to try to find out at what point do you start seeing Neanderthal genes in what you would consider Homo sapien fossils.
What do you think about one genome or the 3 genomes that they did? Is that enough if they were siblings and family? Because this is, I mean, we're making assumptions about, you know, major species, interspecies, cross-species, you know, exchange
based on bones
DNA.
from a single cave, right? And 2 of the bones were essentially maternally related. I mean, yeah, exactly. You may have picked on one tribe that was especially kinky. I don't know.
Good point.
The thing that comes to my mind is the quality of the samples that were used.
Terrible.
Yeah, and so I mean, I don't know if there are any examples of these amazingly well preserved corpses, if you will, of Neanderthals. These individuals that were frozen in ice.
No, the best they have are the bog people in northern Europe. I think they're what.
Svante told us he was using bones from museum pieces, and that, you know, they were drilling bones.
Right, but my question is, I'm wondering if it might be a better sample to take would be something that was, incredibly well preserved. Either these individuals that were in the bogs, or some of these individuals that were frozen at the top of the Alps, or something like that.
Well, the problem is that you could get them for let's call them young caveman, like the guy they found in Europe. But the thing is that human population did not tend to inhabit these very cold, very inhospitable climates until much later in human evolutions. First of all, we did not have the population to justify going to this hellhole. And the second point is that it still needed a certain level of technology, even if it was very simple technology, to be able to adapt to the kind of climate where you would end up being frozen for thousands of years and then discovered. And finally, the last point would be, well, I mean, like I said, you have to find the fossils in these areas. These are not areas that are easy to explore or dig, and you may get lucky, which is what happened with
sure.
I think it was in Austria and Italy. Did they even figure out whether it was in Austria and Italy that they found this guy? I think it was Italy, but I'm not sure.
I don't know.
Yeah.
But hey, Andre, this is a brilliant story in that, one, it tells us a lot about potentially who we are, but like in all science, it raises more questions than, every time you do an experiment, oh my God, there's 10 more to do, right?
Yeah, but again, you can't help but think that you tend to think of this event as something that happened quickly, and it did not happen quickly. It happened over 30 to 50,000 years, which is 10 times longer than our entire history since the first city was founded. It's very, very hard for us to just grasp the amount of time that it took for essentially Neanderthal to disappear.
Maybe also one thing that we can be reassured is that it probably wasn't a single time event, because what's the likelihood that we find the bones of a Neanderthal family, that is the one, right?
Well, their statistics is actually quite good in the sense that you're looking at the similarities between the genome and what's the possibility that this would occur by chance, and it is their p-values are excellent.
Their p-values?
Well, sorry. It's a measure of statistical significance. I mean, if you see essentially that this human looks more like just the non-African genomes and not the African genome, what's the possibility of this happening by chance? And it clearly did not happen by chance. Now, this whole hypothesis will be tremendously reinforced when somebody, in my book, essentially gets Neanderthal bones from other sites other than Croatia. And even better, if they take bones from very early Homo sapiens that have moved into Europe and find Neanderthal gene sequences in them, then I'll be convinced.
Well, I'm sure they're thinking about this down the same lines, right? Svante is a very, very smart individual. He's doing really great work, and he's got a great team of people around him. So maybe we should have him back on as well to discuss it further. He's got a great sense of humor too. He's fun.
They've got the methods figured out now. So once you figured out the methods, it's a lot faster to do more samples. And I bet a lot of paleontologists are going to be shaking bones at these guys and sequence that one, sequence that one.
I would
shake a bone at it.
Yeah, well, it's going to cost $7 at one point. So here's the segue to the last story. And the last story, I'll just present it because it was a paper that came out yesterday, and I read it at midnight. I found it at midnight on Dig, and it was the story of the first organism engineered where its genome was completely synthesized in vitro by machines that just pump out DNA. And what's interesting about it, and how it bridges to your story, is that I asked Drew Endy, who has a company with George Church, and what they do is they make DNA and they make genes, they make synthetic genes. And I asked him, okay, well, now that we have the human genome and we'll eventually have the Neanderthal genome in full, will we be able to synthesize these and create an organism with the Neanderthal genome? And he said, well, right now it's very, very, very expensive. But in 20 years, why not? It'd be really, really cheap. The price, and he did the economics of the price drop. So we're potentially looking at, based on the paper that I'm presenting today, a future within 50 years where we could synthesize the Neanderthal genome in its entirety and throw it into a human, egg that where the nucleic acids have been, cooked out. So
I think we'll do a mammoth first.
Mammoth first. That would be more fun. And think of the
cool.
think of this. Yeah. I was just thinking amusement park you could have. It'd be fantastic. So so that raises the ethical issue of this was Craig Venter's paper from the Craig Venter Institute. Okay, so just to put a context onto this, it cost $40 million. It took 15 years for 20 scientists to do. And what they did is they made an organism, a mycoplasma, which is a really small bacteria. But what they did is they synthesized using machines the DNA completely chemically they used chemicals on the bench, made DNA. And there's DNA machines. If you want to listen to Lee Hood, who invented the DNA synthesizer, we had a Futures in Biotech with him. Brilliant man. That was the early episodes between 10 and 20, somewhere in there. He's been on a couple of times. Great guy. So here they're using their machines. They're synthesizing DNA. What they did is they assembled 1,000 base pair segments, and then chemically. Then what they did is they pooled them together using yeast as a tool. Yeast have the incredible ability to fix broken DNA. And so what they did when they synthesized the DNA, they made overlaps that look similar. And then the yeast pops these together like Legos. So they built, they took 1,000 bases, then made 10,000 base pieces in yeast. Then they made 1,000 base pair yeasts, or base pair pieces. And these, 1, 10, and then 100,000 base pairs. And then they took 11 of those huge DNA constructs, developed the technology for doing a lot of this manipulation, and then one at a time swapped them into a genome that was similar in the yeast, right? And then the yeast has the ability to fix it and swap them in and swap them out until they had completely replaced the original genome with completely synthetic fragments. Now they had the first organism, and they call it the Mycoplasma mycoides JCVI SYN1 genome.
Venturi.
Pardon?
I'm joking.
They call it the Craig Venter bug. Anyway, so they completely synthesized an organism, or not completely synthesized an organism. They have there's a living organism on Earth today whose genome started in a machine, not in another relative, in the mother-daughter cell pathway.
Synthetic genome.
And I'm going to ask the panelists, and I'm going to ask the non-geneticist, so Dave, what do you think?
Well, I think, how do you mean? I mean, it's kind of...
Well, do you welcome the new synthetic organism overlords?
Well, I mean, to me, I think this is going to lead to treatments for a lot of things. I mean, when we figure out the genetic basis of a lot of, say, let's think about something like a psychological disorder like schizophrenia. If we can do that and we can figure out what the genes are, we can, I mean, this is a long time in the future, swap them out, right? So, I mean, I actually don't have a great deal of, I think science is amoral anyway. I think it's moral and immoral what people do with it. So, I mean, I think something bad could be done with it, yeah. But to me, well, first of all, it's just, it's kind of gives you shivers up and down your spine if you like science, right. Because it's like that's so cool. On the other hand, though, I mean, excuse me, are there potentially bad possibilities here? Of course, but I'm not. Like I said, there's also the possibility of helping people, and of course of perhaps building really cool pets. So you got that going for you.
Hey, well, so Vince, do you think this was a major paradigm shift in our abilities as genetic engineers?
No, I think not a major paradigm shift. I think it's a technical tour de force. It's amazing to be able to make a 1 million base genome and assemble it. It's amazing to be able to put it into another organism. But what do you want to do with this? I mean, if you want to have the organism make specific gene products, we can already do that. We can introduce individual genes. So if you want to make something completely new, there's a long way to go before you can do that. So I think, technically it's very interesting, but I think the hype machine behind Craig Venter is pushing a lot of this forward where it's not ready to go anywhere yet. So I would echo the sentiments of, let's say George Church. He says, Dr. Venter is not necessarily on the path to produce useful microorganisms. And your friend there, Leroy Hood, said the report is glitzy, but we need to understand lower-level genes and networks before we can try to make an organism from scratch. So pretty technically amazing, but not a paradigm shift, I think.
Well, the organism that they created, right, it has 485 protein coding genes. So it's extremely, extremely small, 100 of which are not essential genes. You can just take them out. So the organism could potentially live on 385 protein coding genes. So it's not like they built a mammoth yet or a Neanderthal, but, you know, what's Venter's next step, right?
Well, these are the initial sort of baby steps, I think you would say. In terms of practical uses, I would definitely want to echo, you know, what Vincent was saying, which is, so what are you going to use this thing for? And I think initially, short term, you know, most of the things that we can think of, that we can envision, can already be done in multiple other organisms. But long term, if you look further into the future, I think you see that this is sort of a baby step towards bigger things. What those bigger things are, I'm sure Venter will tell us in a couple of years.
It could be important, but look, we have how long has life been on Earth, right? 4.5 billion years, maybe 3 billion years. It was 1 billion years without life, the Earth, and then evolution has pushed organisms to use resources, O2, CO2, water, and sun, in every possible angle. And so no matter where you look, there's life on Earth and organisms capable of doing chemistry that is incredibly broad. So maybe, you know, rather than if we want oil-producing bacteria or oil-consuming bacteria, maybe there's an oil-consuming bacteria out there. You just go look in the around the oil slicks in the tar sands of Canada, and there's bacteria that are just thriving on it. Who knows? Sure. Maybe then take it from there.
Or there's some sort of organism out there that's adapted to a similar type of environment and just needs to be tweaked a little.
Yeah.
You don't have to remake its entire genome in order to get it to do what you want it to do, because this is biology engineering.
Well, just programming a genome from scratch to do it exactly what you do would be tremendously complicated. I mean, just for us trying to figure out what a few genes actually do in a context of a systems organism. So it's already there. We're trying to understand what it does, and we can barely do it as it is. And essentially what they're hoping to do is they're essentially say, okay, I'm going to start from scratch and create an organism that will eat oil and that will not kill every living plant on Earth by accident, essentially. And that almost happened.
I'm glad you're thinking about it, Andre.
Yeah, so I mean, just the amount of work involved to initially design this organism. Now you know it can be built, but it will take a long time before somebody can say, I'm going to start from scratch and design the oil-eating, the let's call it the Louisiana sweet crude eating organism that I can just pour in the Gulf and solve my problem in a week. We're not there yet.
Since there's quite a few authors on the paper, there's probably one bound to be a Fib listener. So I'll congratulate you guys on the paper. There's some brilliant techniques that you guys did and developed on how to isolate these large bacterial constructs, where they're yeast constructs. They're made in yeast, and some very, very elegant technologies. And congratulations on that. So let's see where it goes. Hey, I'll go around the table here. I'd like to thank Dave Broadbeck, who's Associate Professor and Chair of the Department of Psychology at Algoma University. Thanks, Dave, for coming on the show.
Always a pleasure. And I just wanted to mention that on June 18th, this sounds like I'm leaving the Johnny Carson show, but from June 18th to 20th, I'm going to be at a conference called Podcasters Across Borders, appearing on a panel with Twit's own Tom Merritt.
Oh, cool.
Yay.
Yeah, so I thought I'd just mention that.
Cool. That's really neat. Yeah, and you can visit him at thunderbird6.org. He has
that's one place.
Yeah, he's got a network himself.
And you can also get my lectures, mostly sort of neuroscience and statistics. That's people.auc.ca/Broadbeck/blog.
Cool. Great lectures, by the way.
Thanks.
And go Habs go. So
indeed.
On that same note, go Habs go, we have Andre Nantel, Senior Research Officer at the National Research Council of Canada, and he's also an adjunct professor at McGill.
They keep switching to
on the screen. There we go. Now we've got Andre on the screen. Thank you, Andre, for coming on the show. Great story and great job on it.
Go Habs, go.
You can also see him at digitalapoptosis.com. So digital is pretty easy to spell, but apoptosis, APOPTOSIS.com. He's a fantastic photo blogger. I check out your site every day. You probably know that.
You all should.
Fantastic photos. You travel the world and you share it with us. So thank you for doing that. We also have Vincent Ragnialo, who's a professor of microbiology and host of This Week in Virology, host of This Week in Parasitism, 2 great podcasts, science podcasts by scientists and really great professors. And you're a professor in microbiology at Columbia University. Thank you for coming on the show. You did present a crazy story that could save 200 million lives, and I think that was pretty brilliant.
Mark was great, and I really liked those other stories today. I really learned a lot. So all around, it was great.
Well, thanks a lot for coming on. And last but not least, we have George Farr, who's Vice President of Biochemistry and Biophysics at Aeromics LLC in Cleveland, Ohio. He's also an adjunct professor in the Department of Physiology and Biophysics at Case Western Reserve University. Thanks, George.
Thanks a lot.
Thanks
everybody. It was
thanks
fun.
for not playing footsie with me here.
It's like the Bobbsey twins. It's ridiculous.
Right. For the people in the listening audience, we're side by side here on one camera, so it would simplify some of the engineering.
But thanks everybody. It was a great time.
It's always fun. So let's do this. Let's do this again, and keep your eyes open for the great scientific stories, and keep looking forward. So, before we go, actually, there's a couple more people I'd like to thank. I'd like to thank Bert McQuinnan for handling the audio and video boards and recording today. I'd also like to thank the team that make this possible: Leo Laporte, Dan Golden, Colleen Kelly, Eric Lanigan, Tony Wang, Frédéric Louis, and the rest of the TWIT team in Petaluma, California. Lastly, I'd like to thank Phil Peltier and Will Hall for the opening and closing themes. For Futures in Biotech, I'm Mark Peltier.